Hi class, and welcome to chapter 1 in our Physiology at Rio Hondo College. This is an introduction to physiology chapter. We'll be going over some of the tissue types that you might have learned in anatomy or you should have learned in anatomy. So I might skip over some of those that you learned in anatomy, expecting you to remember that, but we will go over more of the physiology-based slides in this chapter. So what is physiology? So you just finished up anatomy, and anatomy was the study of biological structure. So you learned the names of things. Well, physiology is the study of function, now how all the body works together. So we'll look at this course, the function of cells, we'll look at mechanisms, we'll explain cause and effect sequences of the body systems. And we'll look at how how the body works has been learned and derived from different scientific experiments. Pathophysiology looks at how disease or injury affects physiological processes, which would aid in understanding normal processes. And comparative physiology would look at the differences and similarities in the functions of invertebrates and vertebrates. So you can kind of compare two species with each other to learn about physiology in general too. And this has aided in the development of pharmaceutical drugs, especially testing a drug, for example, on lab animals. So in order to understand how we kind of get to how we've learned about physiology is everything has gone under the scientific method before. And I hope this scientific method or the idea of it is a little bit of a review for you. The steps of your scientific method include making an observation or identifying a problem or something that you find interesting in the world around you, and then forming a hypothesis. A hypothesis is kind of a guess or an explanation to that problem or observation. That hypothesis must be a statement that is able to be tested, because then you'll design and conduct experiments or make more observations. You'll analyze and collect the data from your experiment, and then you'll be able to look at the results, and you might get lots of different results many times before you can establish any sort of conclusion. And several verified hypotheses after you do the experiment over and over again, if you keep getting the same result, that could become a general theory. Measurements, controls, and statistics are all a part of this process too. Good physiological research requires a quantifiable measurement that you can measure, an experimental group, and then a control group, and usually they don't know who they are. Statistical analysis, where you use a lot of math to determine if something is statistically relevant or not. And then the review and publication by a peer-reviewed journal, where you would have other scientists look at your experiment and tell you if it is legitimate or if there's any problems with it. So this is especially important in physiology when we talk about developing pharmaceuticals, or let's say a vaccine. Basic research is always conducted for years before a drug is ever given to a person. Research begins by looking at the effects of a chemical on cells in vitro, which means in a cultured disc. Next, studies are usually done in rats and mice. That's a common lab animal because they can reproduce quickly. So you always have many of them to work with, to see if the same effects occur in vivo, in a living creature. So in vitro is in a cultured dish, and then in vivo is in a living creature to see if there are any toxic side effects of the drugs. For these trials, many rats and mice are genetically modified to be susceptible to a particular disease, and animal trials could take several years before a drug is actually approved. Phase 1 clinical trials will test the drug on healthy human volunteers to test for side effects, rates of passage, doses, etc. Then phase 2 would test its effectiveness on people with the particular disease. Phase 3 would be conducted on a large number of people to include both sexes, age groups, ethnicities, and people with more than one health condition. And from here, in phase 3, the FDA could approve the drug for sale, but then phase 4 would even test other applications for the drug. So homeostasis and negative or feedback control are very important with both anatomy and physiology. And I'm again hoping that these two words are a little familiar, because I'll go over them a little quickly because this chapter is a little bit of piggyback off of some anatomy concepts. Here's a brief histology of physiology. I'll let you go read through this on your own. taking us through Aristotle, William Harvey, Claude Bernard, and eventually to Walter Cannon, which coined the term homeostasis to describe what we know as the internal consistency or maintaining a constant internal environment of the body. And that's what homeostasis is. And all of our physiological mechanisms' main purpose is to maintain this constant internal environment of the body. That means pH levels of the blood, respiratory rate, temperature of the body. Any sort of deviation from homeostasis could indicate a disease or disorder. And most homeostasis is accomplished by negative feedback loops, where we have a sensor in the body that detects a change that's out of normal range. That sensor would send that change or that information to the integrating center, which will assess the change around the set point. The integrating center then sends instructions to some sort of effector, which will try to make an appropriate adjustment to reverse the change back to the set point or back to normal. So mechanisms of negative feedback are to move in the opposite direction from the change, because if the change is really hot temperature, the negative feedback wants to bring you back to normal. So that's what means is going in opposite change. It also tries to make the change from the set point smaller, reversing the change. And this is always a continuous process. It's probably going on right now in your body, always making little fine adjustments so that the inside of your body is always around normal parameters of all the variables. And your first lab, I think, or your first or second lab will go over these negative feedback loops and how maintaining homeostasis is important. How you have kind of a normal range, and if anything gets out of that normal range, a sensor will be activated, and then an effector will be activated to try to bring, let's say, temperature or pH back into normal range. Body temperature is kind of a big example of this, so I'll take you through body temperature as an example of negative feedback. For example, a sensor in the brain would detect any deviation away from 37 degrees Celsius, which is normal body temperature. And then another part of the brain will assess this as an actionable item. And effectors like sweat glands are stimulated to cool off the body. Once your body is cool, sensors will then alert the integrating center in the brain, and then the sweat glands will be inhibited once we bring our body temperature back to normal. And the end result regulates the entire process, where once your body temperature is back to normal, the end result will turn off then that process. Production of the end product will shut off or down, regulate the process, and this is why we call it a negative feedback loop. And antagonistic effectors describes how homeostasis is often maintained by opposing effectors that move conditions in opposite directions. So this maintains conditions within a certain normal range, which is what we call dynamic constancy. And when you are hot, you sweat. When you are cold, you shiver. And these are antagonistic reactions. Other examples of variables that will change with antagonistic effects or opposite effects would be controlling blood glucose levels within normal range, blood calcium levels, your heart rate, and blood pH. So again, this is just a look about how sweating and shivering are antagonistic effectors, meaning what they do, sweating versus shivering. Sweating releases heat to try to cool down your body, and shivering tries to create heat to warm up the body. So those two effectors work in opposite to try to keep your body temperature always at normal range, which is 37 degrees Celsius. Quantitative measurements. In order to study physiological mechanisms, scientists must measure specific values and then mathematically determine such statistics as their normal range, their averages, and any deviation from average, which could be the set point. So a knowledge of these normal ranges will aid in diagnosing a disease and assessing the effectiveness of a drug and other treatment. So these are just approximate normal ranges for measures of some fasting blood values. So you usually take a blood test at a clinic when you've been fasting so that your food doesn't affect your blood sugar or your temperature or anything like that. So these are just normal ranges of pH, bicarbonate ions, sodium, calcium, and the rest. And again, if anything is out of normal range, your body will compensate for that, or that will be indicative to medical health professionals that there could be a disease or a disorder that your body is fighting. Positive feedback is where the end product in a process will stimulate the process in the same direction, so the action will amplify the change that stimulates the effectors. Positive feedback could never work alone, but it does contribute to many negative feedback loops. So for example, if you get cut and a blood vessel is damaged and you're bleeding out, a process has begun to form a clot. And once the damage is fixed, clotting will end. However, the process of forming the clot involves positive feedback to bring as many platelets to stop that blood as possible. Also, a big one is in uterine contractions right before childbirth. This is regulated by a positive feedback loop with the hormone oxytocin, where the levels of oxytocin continue to increase to stimulate stronger and stronger uterine contractions until the baby is born, and then it shuts off. Regulation of processes within organs can be intrinsic or extrinsic, where intrinsically cells within the organ will sense a change and signal to neighboring cells to respond appropriately, or extrinsically, where the brain or other organs will regulate an organ using the endocrine or nervous system. The nervous system will innervate all organs with nerve fibers, and the endocrine system will release hormones into the blood, which can transport them to multiple target organs. So this is just looking at how your bodies are regulated with neural regulation, nervous stimulation from the brain and nerves, and also endocrine regulation, where different hormones will be released to try to keep everything back to normal as well. Hormones are always secreted in response to specific stimuli. So an increase in blood sugar will result in insulin secretion to try to remove sugar from the blood. And secretion can be inhibited by its own effects, where a decreased blood sugar will inhibit the release of insulin because you don't need any more insulin to decrease the blood sugar at that point. And we'll talk a lot about your endocrine system when we get to that chapter. Negative feedback inhibition usually involves an antagonistic effector to make sure that homeostasis is maintained within that normal level. So for example, when blood sugar is low, the hormone glucagon is secreted, which will result in a rise in blood sugar. So glucagon is the opposite hormone from insulin, where insulin decreases blood sugar, glucagon actually increases it. So again, these antagonistics, glucagon and insulin, work to do the opposite functions to try to keep blood sugar within normal range. And that is shown here, where eating will increase blood glucose. So then you'll have your cells in your pancreas releasing insulin, so that cells will take up glucose to decrease blood sugar. But if you're fasting, when you have very low blood glucose levels, you'll have cells in your pancreas that will decrease the secretion of insulin and increase now glucagon. And what glucagon does is it tries to put glucose back into the blood to try to increase your blood sugar levels. And again, once your blood glucose levels are back within normal range, that kind of end result will then turn off kind of the first kind of stimulus or the effect that's being produced. And that's where this negative sign is showing that the end result, once it's back in normal range, that will turn off the initial stimulus, and that's what the negative feedback loop is. Okay, so this primary tissue section you are responsible for, but this is a huge review from anatomy. You should know these levels of organization. I love to test on this to make sure you know what the basic unit of life is and how cells come together to form tissues, organs, organ systems. We have 11 of them, and then the entire organism. A lot of probably the next 15 slides I'm going to skip, but you are responsible for them, especially understanding the 4 major tissue types in the body and how each tissue has a particular function that will dictate the physiology of the organ. You should know the 3 different types of muscle tissue and how they're specialized for contraction. We will go over how they contract when we get to the muscle tissue or the muscle system, but I do want you to review kind of the basics of skeletal muscle, how it looks, the basics of cardiac muscle, how it looks, and the basics of smooth muscle. So again, This should be a review from anatomy. If it's not, I need you to kind of study these slides on your own because you should have learned it in anatomy. Nerve tissue is found in the brain, spinal cord, and nerves. So you should know well the anatomy of a neuron. And we'll learn when we get to your nervous system chapter, then the physiology of how the neuron works. And then epithelial tissue, how epithelial tissue is named. So here are all the types of epithelial tissue, their structure and function, and where they're located. So you should review this slide as well. So I'm going to skip over this, not to be mean, but as it is a review from anatomy, and I want to keep, in general, I try to keep my lecture videos between 30 to 45 minutes to try to encourage you guys to watch the whole thing. Connective tissue is the most diverse tissue. And again, you should review the structures of connective tissue. Okay, and then organs and their systems. So again, tissues come together, more than one, to form an organ. And an organ is composed of two or more tissues that serve different functions of the body. The skin is your largest organ in the body. It makes up about 15% of your body weight, and the skin has all four primary tissues. The epidermis is made up of an epithelial tissue. The dermis, we have different types of connective tissue. Your hypodermis is made up of adipose tissue. So that is your skin, and you can see the skin here, and we'll have some more information when we get to the integumentary system. But your skin, you have the epidermis made of epithelial tissue, which is the covering layer of the skin. The dermis is made up of connective tissue, where we have all the hairs, some erector pili muscles that give you goosebumps, the different types of glands. And then the hypodermis is made up of kind of this adipose tissue of fat, insulating layer to help keep our bodies warm and protect the organs that lie beneath it. Some tissue development, all tissues are composed of highly specialized cells that arise from these three tissue or germ layers in the womb during embryonic development. The types of cells, types of stem cells, a stem cell will be a cell that can go on and differentiate or specialize to be a different type of cell. Zygotes are totipotent, and a zygote is the name for the first unifying or fertilization of a sperm and an egg. So zygotes mean that their cells can become any type of cell, and these are true stem cells. And as stem cells, or as these cells begin to differentiate or become their specialized type of cell, a few adult stem cells will be retained to allow for cell replacement. Adult stem cells are still limited to a narrow range of possibilities, but they can still become several related cells, and thus are called multipotent. So for example, a bone marrow cell can become any type of blood cell. So you'll often hear of someone with cancer or other diseases having a bone marrow transplant, because a bone marrow transplant of bone marrow cells can go on and become extra red blood cells or white blood cells that diseased or patient person might need. Embryonic stem cells are called what we call pluripotent, where they can form any type of unrelated cell. Here we have stem cells in a bulge, for example, of a hair follicle. They're found here in the lower kind of bulge region, and they'll go on and form the cells that will eventually form the hair follicle itself. Organs then that perform related functions are grouped into systems, and we have 11 interrelated organ systems in the body. You should review these organ systems of the body. You should have a very good understanding of the main organs in these 11 organ systems, and you should review what their primary functions are. This, again, I'm hoping is a review from anatomy. And then throughout this course, we are going to be going over individually what happens on a physiological level in your respiratory system. So gas exchange, endocrine system, hormone release, reproductive system, GI system. So now we're really going to get into the nitty-gritty physiological functions of all of these organ systems. And that's what makes physiology so fun, is you've learned the anatomy. You probably remember what all these organs are and the general function of your body systems. But now we're going to look at how things work on a cellular level, which will be very fun. Or maybe that'll just make me a nerd, but it'll be a great course. Body fluid compartments, just a little review here. If we say something is intracellular, that means anything, any area inside the cells. And inside your cells contains about 65% of your total body water. And if we describe something as extracellular, that's the area outside the cells. This would be all your blood plasma and interstitial fluid between the cells. Both body fluid compartments are filled primarily with water and are separated by membranes. And that's the end of chapter 1. So this is a brief introduction to physiology, but I do want you to take the time to review all the anatomy as well. That is found in this slide because you're responsible for all the material in a PowerPoint, unless I specifically tell you not to worry about it. So we'll see you for chapter 2. Look for that recording being posted soon. And I hope you guys are all staying healthy and well, and take care.